My name is Camille Williams, and I would like to present to you the history of CRISPR and also the medical uses of CRISPR-Cas9. The article that I reviewed in connection with this presentation was by Eric Lander, titled The Heroes of CRISPR. The citation I chose to follow for this lecture is it basically tells about the medical uses of CRISPR and also the history of CRISPR. CRISPR is an acronym that means clustered, regularly interspaced, short palindromic repeats. The name can be broken down into two parts. The first part being the CRI, clustered, regularly interspaced, which basically means that the DNA that repeat are interspaced. And the second part, SPR, short palindromic repeats, meaning that there are short segments of DNA that repeat that are never odd or even. In the middle of these DNA, where the spacers are, is where spacer DNA is contained and is never identical. The spacer DNA are the CRISPR-related DNA, and they're called Cas genes. The Cas genes can cut or edit DNA. So, if there was some fault in DNA, like a disease for instance, that part of the DNA could be edited so that the disease is not present anymore, which is life-changing. The first characterization of CRISPR took place in 1993 by Francis Mojica. By the year 2000, Francis Mojica was able to realize that what he had originally thought to be disparate sequences that repeat turned out to share commonalities. These commonalities are what are known now as a trademark of CRISPR sequencing. After that, in 2005, he found that CRISPR is an adaptive immune system. In 2005, it was also noted that the Cas9 gene had a nucleus in large protein. In addition, the protospacer adjustment motif was also discovered, and it is called PAM for short. PAM is needed to find the target that would need to be edited. In 2006, it is noted that Cas9 can be used on novel viruses as well as viruses already known to an organism. In 2008, scientists recognized that the target molecule was DNA, not RNA as previously thought. In 2010, Cas9 is recognized as the only protein needed for cleavage, specifically in the CRISPR-Cas9 system. This feature is unique to type 2 CRISPR systems. In 2011, researchers found that there was an additional type of RNA present with CRISPR RNA, which is called tracker RNA. These two types of RNA together guide Cas9 to its target. Also, in 2011, researchers were able to signify that CRISPR systems are self-contained with type 2 systems by using the CRISPR-Cas9 system in E. coli, which is not a type 2 system, and CRISPR provided plasmid resistance. This provided that all of the type 2 systems were known. In 2012, one of the biggest developments that was made with CRISPR was that Cas9 could be reprogrammed so that scientists could choose a site that they wanted to be edited by editing the CRRNA, the CRISPR RNA. In 2013, researchers were able to target genome cleavage in human and mouse cells. To understand CRISPR as it is today, Scientists had to understand how DNA works, the concept of genes, and the function of different parts of cells, just to name a few concepts that had to be thought of and mastered before CRISPR could even be a thought. CRISPR now has profound impact on how we think about cures for diseases in the future. Scientists have found that the impact it would have in pulmonary and gastrointestinal diseases would prove to be monumental. As a homozygous mutation in the cystic fibrosis transmembrane conductance regulator gene was corrected using CRISPR-Cas9. Additional uses of CRISPR-Cas9 in medicine is the use of CRISPR-Cas9 in breast cancer research, Hirschsprung disease, sickle cell anemia, HPV, HIV, HPV, and many more diseases and disorders. This visualization of CRISPR shows a CRISPR molecule finding a precise location in the target DNA. Then, the CRISPR enzyme cuts the target DNA at the point found by the guide. Then, a new custom sequence can be added when the DNA is repaired. The primary uses of CRISPR technologies are used for medical uses. While gene editing can target a wide variety of DNA and RNA to even genetically modify babies, medical uses are at the forefront of current research.